Well, it start with the medicine and physiology, then physics. The third one is chemistry. Fourth one is literature. Fifth is fees, and finally the sixth one is going to be the economics. So we used to conduct lectures in first three natural sciences, particularly medicine or physiology, physics. And today we are having the lecture on Nobel Prize in medicine. Tomorrow we have the lecture on Nobel Prize in physics, and the Nobel Prize in chemistry is going to be held on 2nd November. In the meantime, we have one more PSL on 26. So before introducing starting the meeting, we have, I would like to point out a couple of things about the PSL and Tamil Nadu Science Forum. So PSL is part of Tamil Nadu Science Forum. Tamil Nadu Science Forum was founded in 1980s by few research scholars. One among them is our Dr. Professor T.R. Govindarajan, who was doing his PhD in Madras University. And those research scholars joined together and are form a Tamil Nadu Science Forum. Initially, they were conducting hall meetings to the public to popularize the science. So they conduct a lot of meetings. At some point in time, they decided to go beyond hall meeting and go to the public. That means they go to the places, residential places, and start giving science lectures. Then they develop a program of going for a science yatra. It has happened from Kanyakumari to Chennai in 1986. The response they received led to the formation of the Tamil Nadu Science Forum as a state level unit. It was initially an organization confined only to Chennai. It becomes an entire state. Now we have the Tamil Nadu Science Forum is functioning throughout the state. This is one interesting thing. Well, once, as you know, that when the quantitative change happens, qualitative change will also happen. So initially we gave lectures. Now the activities has spanned into 6 different areas. One is the science publication. The second one is the science communication. That's what initially we founded. The third one is the youth for science, and fourth one is the women in science, that we have a separate group working on it. It's called Samam. And the fifth one is we work on the environment and the development sciences. And the sixth one is the education. So there are 6 different areas. I would say that 6 different verticals. Tamil Nadu Science Forum is working on it. The state level body, we have 6 different subcommittees. We have district level bodies, we have 6 different subcommittees. The important thing is that now in science publication, we have been publishing 2 important magazines. One is Tholir in Tamil, which is meant for children. And the second one is in Jantar Mantar, that's in English, meant for the college level students. These 2 magazines are running. We are publishing from 1987 onwards. Okay. If you look at the magazine, no, we don't have any advertisement at all. It is surveying on its own strength. This is an important thing I would like to say about the Tamil Nadu Science Forum. Apart from that, when we used to have the conference once in 2 years, and we find out that we should work more on the higher science. That's how the PSL has come into picture. From 2080 onwards, the PSL has come. We started giving the lectures using PSL. This is the 82nd, 81st PSL, and we'll be having 82nd tomorrow, 83 on 26, and 84 on November 2. So this is how. This is a small introduction I can say about the PSL and Tamil Nadu Science Forum. I would invite Professor Govindarajan to say something about the PSL and introducing the speaker, Professor D. Karunagaran.
Thank you. Nandri. I request some of the students probably to come forward. That may help. That's no compulsion, but please, for your own benefit. Okay. Tamil Nadu Science Forum has been, as he mentioned, in existence for the past 44 years, from 1981 onwards. Unfortunately or fortunately, I was one of the person involved in even starting it. So it has grown to a very big organization with more than 20,000 members, various districts all over Tamil Nadu, we take science. The whole idea is to take science to all sections of society, particularly school children, colleges, and even people who have quit or left and gone to jobs, and science lectures through some of these things. So this is part of the program. And as he mentioned, we have the magazine Tulil and Jantar Mantar, and we also have Children's Science Congress, etc. Now we want to go into newer areas. With that in motivation, for the past from 2018, we started this popular science lectures. So this is 81.
81.
This is 81st lecture. So even during COVID, we had online lectures. So several lectures were organized, and I must tell you, I gave the talk in this same hall. The first talk, well, I gave the talk, and this hall was full. So now many people, since it is available in YouTube and other things, people who register with us, some of them prefer to listen to the talks through YouTube and or probably say later, but they are going to miss one thing. You are not going to interact with the speaker, right? You are not going to interact with the speaker, and coming here is actually beneficial. And probably that message should go to the all sections. That COVID forced us to do online lectures. So, but now we can move forward to offline lectures. But COVID also provided us this online mode, which makes this record available for future generations for many times. So that is good as far as thing is concerned. So this is today's, we have been conducting 2018 onwards, this Nobel lectures every year in October. So 3 lectures will be held: physiology, physics, and chemistry. And once we held on economics also, for the climate change issue, because it was also related to scientific question. Yeah. So that was a kind of a thing. So except for that, we have always held only physiology, physics, and chemistry. Today is going to be physiology about miRNA, which got the prize, and our speaker, Dr. Karunagaran, will explain. But tomorrow is going to be on physics. In fact, one of my colleagues is going to speak, and another from IIT Madras is also going to speak. So this is the program for physics tomorrow. And as you know, this physics this time is for artificial intelligence. So it might be more interesting. Some of you who are interested can join. On 2nd November, we have the chemistry program. And again, so turned out there is some mix of artificial intelligence and machine learning. So some of these things are new things which our younger generation, younger people should learn, at least the words, and move forward, because the world is going faster than we can imagine. And lastly, this time we, in a sudden burst of thinking, we introduced a competition for all the registered people to predict the Nobel Prize winners. We started only 15 days before. We informed to all the sections of people who were registered with us earlier, and we did. Our response was, because it was too late, was a little poor, but we were not expecting anyone will predict successfully. It so turned out, partly it was true. There is no prediction in physiology. There is no prediction in physics, but there was a correct prediction in chemistry. So that was an interesting thing, which we should acknowledge. And this is with a small number of participation itself. People can, of course, now with ChatGPT or AI, Gemini, no, we are not stopping anyone. You can ask anything, anyone, anybody can be consulted and come with some name and what is the area. Okay, so that is the interest we wanted to create among the students and young people going to colleges and other places. And hopefully next year, we will start it sufficiently ahead, more than month or so ahead of time. So it becomes very popular so that we get to thousands of predictions. And of course, such predictions in large number will always result in some good predictions also. Even in this thing, about within 100 people who are predicted, one was correct. So, and accurate. It is the name of the person was predicted correct. Yes. And that is an important thing. We are going to give the person a prize, thanks to IIT Madras, who have sponsored part of the prize money. And we are going to do that on 2nd November. So that is for the conclusion. I don't want to take too much time. Dr. Karunagaran was the Emeritus Professor from IIT Madras on Biochemistry and Biotechnology Department. No, it's okay. And he is well known for his work in this miRNA, particularly with respect to how one can deal with cancer, particularly breast cancer. Am I right, sir? Breast cancer. So this is the thing. And I don't want to, we have already written in our notices some details about him in things, so I don't want to take too much time. So now I would request Dr. Karunagaran to present your talk. Yeah. It will go on about 45 to 50 minutes, and then we will have another question and answer session for maybe even half an hour. Okay? Okay. Thank you.
Sure. Are you able to hear me?
The phone, may I request the phones be kept silent mode, please?
Very good evening to everyone. And I take this opportunity to thank the organizers, especially Dr. Krishnaswamy and Dr. Gondradalu and other organizing committee, Dr. Vijayan and others who have taken the pains to contact me and ask me to give a lecture here. Let me start with this year's Nobel Prize in Physiology and Medicine went to Victor Ambros and Gary Raukin, all of you must have seen, on microRNAs. I will give a brief introduction about microRNAs. And we have done some work on microRNAs, which, if time permits, I will share a little bit, although the abstract says most of the things about our work, but I will try to restrict that to a small portion of the talk. Okay, let me. Okay. So as the slide shows, I am in IIT Madras, because I did my PhD in Karunagaran University, that's what it shows. Yeah. I have to keep it a little bit above, closer, yeah. Now you are able to hear? Yeah, thank you. So my lab focuses on cancer biology, mainly on the aberrations in signaling pathways and then mechanisms of potential anti-cancer agents, and also on the role of microRNAs. When I started as an independent investigator after my postdoctoral studies in the US and Israel, I came back to Trivandrum, where I was working on some of these topics. But then when I joined IIT in 2006, at that time, these microRNAs were becoming very popular. So I started, I was fascinated by some of the results that were being published. Since I had some experience on signaling, I started to work on these microRNAs, how they could affect signaling. I'll come to that. So all of you know and must have heard about human genome. What is human genome? The total genetic content in a human cell, you call it as human genome. If it is in the rat cell, you want to call it as a rat. If it is cat genome, and so on. So it represents the total genetic information. Now, if you take our human body, we have several tissues: brain, liver, stomach and all that. All these tissues have genes and the genome is represented. In fact, all the tissues have the same genome as it is shown here, which essentially means all your tissues have, let us say, 25 to 30,000 genes in a human cell. That's what people are saying. So these genes are present in all the tissues, but then all the tissues are not same. Everyone knows that. That is because these genes are expressed differently, which we call as differential gene expression. So how they are regulated, why certain genes are expressed in certain cells, how that is regulated, all these things put together, they say gene regulation or regulation of gene expression and so on. They have these things. So this has not been understood fully, but this is a very important thing, which essentially means this gene expression is very important. If you change the gene expression, you can actually change the tissue. For example, stem cells, if you take fibroblast cells, from that you can even make brain cells. All you have to do is change the expression of the genes. All of us are born by the fusion of sperm and ovum, and ultimately you call that as zygote. When you look at the zygote at that stage, nothing is clear. It just looks like a mass of cells. But then when it develops into a fetus, you start seeing eyes, nose, all these things. You will be slowly able to make out that, depending on the developmental state. How this, what we call as differentiation, happens, again, because of the changes in gene expression, after all. So gene expression is, since I am a cancer biologist, I used to give this example of cancer cells even. Cancer cells are not alien cells. They are your own cells. They have undergone some aberration to become cancer cells, which essentially means if you change the expression pattern of a normal cell, perhaps you can convert that into a cancer cell. So this kind of theory is there. When you read about how the cancer develops, there are various theories, but many people subscribe to what is called a mutational theory, because people think that there are a lot of mutations that are present in cancer cells, because any type of cancer, there are so many mutations in those cells. So mutation theory is the one that is acceptable to many people. But then there are other schools of thought who say that if you change the expression, after all, it's a developmental phenomenon, you will be able to change a normal cell into cancer cell, although it may take some time and so on. So expression is very, very important. That's what I want you to keep in mind, because for this lecture, that's very important. Can I have the, it's not moving? Is that the next slide?
Yes, sir.
Okay, just go to that. No, previous slide. Yes. I can't operate from here. Yeah. Okay. This I named it as a snapshot of the human genome. If you look at the slide, you will see most of the people, what is a gene? Usually, people think that gene will ultimately make its product. What is its product? Usually protein. So proteins are said to be the workhorses. So they are gene products. That's how people have usually defined for a long time. But if you take the human genome, as I told you, in a human cell, there are about 25 to 30,000 genes. And how many of them actually encode for a protein? You can see that they are translated into protein is only about 1 to say 1.4%. So let us say 2% even, it's okay. But what happens to the 98%? People are trying to call that as dark genomic matter. Sometimes they say that it is a junk DNA and so on. There are various terminologies. This is a very interesting concept. See, as a teacher, I was a teacher actually. I started my career as a teacher in PhD College of Arts and Science, Coimbatore. Teachers have a tendency to look at students. Some are considered as bright students, or some are considered as dull students and so on. But I always thought that everyone has a potential. Same thing here. You think it is a junk DNA, but people have now discovered that it has got a potential to regulate the gene expression. That is what we are coming to. So what was thought as a junk DNA now has many components. One of the components is called non-coding RNAs. So when I started off with the definition of the genome and the gene product, we usually think as a protein, but then the genes are going to ultimately produce RNAs. So any functional product, so even RNA, if it is able to produce or make, then that is also important. All of you know the central dogma of molecular biology where people say DNA makes RNA makes protein. So these are continuous things. So if you make DNA from RNA, you are going to call that as reverse transcription. With these fundamentals, let us move on to the topic. Yeah. As I told you, the junk DNA is increasingly being recognized in certain processes. In evolution, for example, there is a role. And in the post-transcriptional silencing, already before this Nobel Prize, actually in 2006, for discovering RNA interference, all of you must have heard about so-called small interfering RNAs. They got a Nobel Prize. Andrew Fire and Craig Mello, they got a Nobel Prize, which is very similar to what we are going to discuss on microRNAs. After all, they are also small RNAs. So in this discovery itself, people found that if you express a double-stranded RNA, it can silence certain genes, which means it can regulate gene expression. Mostly, they will suppress gene expression. Connect with what I said a few minutes back, that if you are able to regulate gene expression, then you can do many things. You can affect the whole system. Because I told you, from fibroblast cells, you can make brain cells. From normal cells, you can make cancer cells if you touch the gene expression. So anything that regulates gene expression plays an important role in whatever the functions the cells are, living cells are supposed to do. So that is why people are attracting Nobel Prize for such discoveries. People may be surprised that all these things started in plants. Even in 1990, Rich Jorgensen in the University of Arizona, he was trying to inject certain genes which will give this flower, petunia flower as it is called, that purple color. He wanted to deepen that purple color and injected the gene that is responsible for that color. Plant scientist, Mendel was able to say that some traces are there and all that. So based on that background, they injected this gene. To their surprise, they found the injected genes ultimately did not produce the deep purple color that they wanted. Actually, it had white or somewhat variegated, not really getting. So somewhere in between. This phenomenon was called co-suppression, because the gene that they were injecting actually did not produce that. It actually suppressed the whatever color that was there already. So that is how they named this. Next. And as I just mentioned, the previous Nobel Prize winners, Andrew Fire and Craig Mello, they were doing experiments with C. elegans, which is a nematode, it's a worm. So in that, they have introduced a double-stranded RNA. DNA is double-stranded. RNA also can become double-stranded if it can fold itself. And that way it will appear as double-stranded. So they had a sense RNA and anti-sense RNA. They injected that to suppress the expression of some gene, which you can see here. The brown color, if it is there, the gene is expressed. If the color disappears, then the gene is not expressed. So when they put sense and anti-sense RNA, they didn't see any color change, maybe some mild change, but not a drastic change. When they put a double-stranded RNA, the color disappeared. So that means somehow this double-stranded RNA had the property to silence the gene. So silencing gene or suppressing the gene expression is an important phenomenon. So they were a bit surprised. Previously, it was not known, I mean, like, except for that plant experiment that I just described. They have done a very elegant experiment. They have worked with a gene called UNC22. It's a myofilament, it encodes for a myofilament protein. They injected a double-strand RNA. They saw that this gene was knocked down. And once this gene is absent, the worm goes in a twitching mode. So that was an experiment. So they were able to demonstrate, yes, indeed, this gene expression is suppressed by a double-stranded RNA. What I am trying to say is the DNA expression is now controlled by an RNA. So that was the discovery at that time. Now, yeah, this year's Nobel Prize went to these two gentlemen, Victor Ambros and Gary Raukin. As you can see, they also worked with C. elegans. Actually, Victor Ambros was working with what is called Lin4. And this Lin4 was able to control another gene, the messenger RNA, what is called Lin14. Lin14 gene gave rise to a protein. Lin14 gene had a protein product, whereas Lin4 did not have any protein. As I told you, in the initial stages, people were always thinking if you have a gene, there must be a protein product. But this one was not coding for any protein. So they were a bit surprised. Another surprising thing that came out from their lab, both of them were actually working in one lab as postdocs at that time. In the 1993, he discovered, Victor Ambros. By around that time, they were postdocs also, and they became independent around that time. So they found that there are complementary sequences in the untranslated region UTR, as we call 3' UTR of Lin14 mRNA. And they hypothesized that probably this is going and binding there, and this binding is probably doing something to suppress the expression of that Lin14 gene. This is what they published. Next slide. But 7 years passed. This is another interesting thing. Even today morning, because you called me for this lecture, I read one of the articles that Ambrose and Raukin have written, in that they are clearly mentioning that they, especially Ambrose, they were clearly mentioning that they didn't think that this microRNA will be present in other systems, because they were working in nematodes. They were thinking that it will be only present in, it's a nematode specific RNA. That is what they were thinking initially. But the same group, kind of Gary Raukin, it was in his lab, he found another microRNA in 2000. 7 years went off. 7 years, nobody bothered about these kind of discoveries. And in 2000, he discovered another microRNA, LET7 microRNA. So that he found to be conserved in all the species. Why one has to bother about that? Because somebody said, I forgot the name of the scientist who said that nothing in biology makes sense without relating it to evolution. So unless you say that it is present in all the organisms, people don't bother much about it. Because ultimately you will be able to help the humankind if you know something, or if you understand the complexity of this regulation in human. So that it can be used for human welfare. So that was the idea. So only after LET7 was discovered, then only people started paying attention, and then a spate of papers came. Remember, I just came here in 2006 from Trivandrum, so by around that time. 2000 they discovered, and then a lot of papers came. And naturally, when we are doing some work in cancer biology, we were also interested to study about this. I will come to that question, why we have to be interested in microRNAs in cancer. Can you go next? Yeah. Then after this discovery, they understood that this is going to be a revolutionary thing. It is going to help in different fields, say development, physiology, or diseases, several diseases, and so on. After all, in biology, the living cells will not do any function. If you take the functions of the cells, proliferation, even cell death, movement, differentiation, neuronal conduction, any function that you can think of for the living cells, they will not be able to do without a signal. So signal transduction is very, very important. These signals will ultimately affect the gene expression also. So in that sense, they thought that if you affect the gene expression and the signal transduction pathways, then naturally it will affect several physiological functions, and thereby perhaps they will be good targets for curing or treating many disorders also. They may explain what is happening in several diseases as well. So that's how it became very important. Next. Okay. The microRNAs, how RNAs are produced, they are coming from DNA, right? By transcription, that's what you call. Similarly, microRNAs also, there are about 2000 to 3000, 2500 or so microRNAs in the humans. There are many others. In the recently with the other Nobel Prize for with the artificial intelligence and all that, the protein structure and all, they have actually, it came in the Hindu 4 or 5 days back when the Nobel Prize was announced, that they were able to find so many RNAs, something like a million, million protein structures they were able to recognize. That's what they said. Previously, we knew only about 45,000 or 50,000 protein structures and so on. Now they are able to. So with the technology, more and more miRNAs are also getting discovered in different species. But in human genome, about 2,500 or so. Okay, they have their own genes, just like any other RNA. They will be produced, and they are also sometimes produced from the non-coding regions and introns also. So like that. But more than 60% of these microRNAs have their own genes. So let us stop with that. And they can be, first they are produced as pre-microRNAs. That is a large, they are usually the mature microRNA is only about 18 to 25 nucleotides in length. That's why they are called small microRNAs, because their length is only about 18 to 25 nucleotides. But initially, they can be produced as a big fragment, and then they are processed and so on. That is what is shown in this slide. Originally in the nucleus, and then they have to be transported out, and they have to come to the cytoplasm, where they have what is called RISC, RNA-induced silencing complex, where they have to interact with proteins and all that. You can see in this slide that there are many components involved in the maturation of microRNAs. Interestingly, once they come out, they can bind with the 3' UTR region of the messenger RNAs. So in this manner, once they bind, once there is a complete complementarity between the microRNA and the sequences found in the 3' UTR of messenger RNAs, then that messenger RNA either gets degraded. If it is 100%, it gets degraded, or the protein will not be translated ultimately. So in this manner, they are able to silence the gene expression. I'll repeat once again. The microRNAs are basically silencers of gene expression, usually. There are some exceptions, but usually they will go and bind with 3' untranslated regions of messenger RNAs and suppress their expression. I told you the importance of suppressing and regulating the gene expression in the beginning itself. I have repeated that. So if you play with gene expression, you can do many things. Just that alone you in mind, then that will help. They will also go and enter into some of the microsomes and can find their way into the fluids also in the system. So exosomes and other vesicles also they can get into. Usually they will go and bind with the messenger RNA and then suppress. Now you can ask, sir, one microRNA, how many genes it will regulate? There are about 500 what we call as target genes. So one microRNA may be able to regulate several genes. That is one thing. There is another complication to this. Remember Watson and Crick base pair rules. A base pairs with T and G base pairs with C. I hope you know that. So based on the same base pairing rules, there is a complementary sequence in the 3' untranslated region of the messenger RNA. If you look at that, immediately it will occur to you, since the human genome is sequenced and all that, you know the sequence of so many genes now, can you find out what will be the probable target if you know the sequence of the microRNA? Do the reverse. If you know the sequence of the 3' UTR, can you find out what microRNAs can regulate? Actually, that is what people have done. There are so many bioinformatic tools. In fact, my own interesting experience is that when I came to IIT Madras in 2006, and IIT students are very good in maths, physics, and all that. They have cleared all these joint entrance exam, these kind of things. So they are good in computational skills, which I am not as a biologist. So I asked one of the students, BTech students, at that time, these computational things were coming. That is, if you know the microRNA, you may be able to predict the target. If you know the target, you can predict the microRNA, which essentially means one target can have many microRNAs theoretically, and one microRNA can target many genes. So that was the precinct with which I started to work. I knew nothing at that time. In 2006, I just heard about microRNAs. I knew nothing about that. Not that I know everything now, but a little bit better. That's all I will say. So I asked this student to come up with some microRNAs that will affect signaling genes, because signaling mRNAs, for example, mRNAs that may ultimately code for some signaling proteins, this, that. So that is what I said. That I said because my experience was in signaling in cancer, aberrations in signaling in cancer. So with that background, I asked him. Next day, or after a few days, he came out with so many microRNAs for a target. So I was puzzled what to work on, because I wanted to work on, for example, I published at that time something on NF-kappa B, which was a transcription factor, and TGF-beta signaling. So I asked him to find some microRNAs that will affect this signaling pathway, so it will be easier for me to do independent research work here in IIT Madras. But then I was really confused, because at that time this was not known. Now I told you that one microRNA can have many targets and one target can have many microRNAs. That point of time, we didn't know very clearly about those things. So I was a bit surprised about this, and basically I was confused. So I told him to randomly choose some microRNA, and then we tried to do several experiments. What we found as the best target based on these sequences was not found to be the best target in vivo. That is also another thing. If you attempt these bioinformatic tools and do some in vitro experiments, in vivo, it could be different. Because the human tissue inside the cells, there may be so many things that are happening. So it may or may not be the same thing. So that is how we ended up with some failures in the beginning, but finally we succeeded in identifying some microorganisms which will affect certain signaling pathways. I will share that after some time. Yeah, next. So why we are interested in cancer biology about microRNAs? That is the next question. Why we should be interested in? All of you must have known what are known as oncogenes and tumor suppressor genes. These are very important in cancer. After all, cancer, people are very familiar with cancer. There is nobody here who doesn't know about cancer because one relative or friend must have got cancer. So some agony is there with everybody about this disease. That's why it is sometimes somebody wrote a book also, Emperor of All Maladies. Siddhartha Mukherjee book. You can read that. It's a very nice book to understand about cancer. Anyway, the point I was trying to say, you have these oncogenes which are growth promoting genes. Tumor suppressor genes, they will suppress the tumor formation. This is another interesting thing in biology. I told you about the signals and other things, signals, factors and all that. Always there will be something opposite, something like your Newton's third law. What is the Newton's third law? For every action, there is an equal and opposite reaction, they say. Here, it need not be equal. It can be unequal or partly equal or fully equal, whatever option you can imagine, these things will be there. Why I am saying, if you take an activator, there will be an inhibitor. If you take a suppressor, there will be an inducer. If you take a factor that will induce cell death, there will be another factor that will induce proliferation. Similarly, you have these oncogenes, which are growth promoting genes, and you have tumor suppressors, which are suppressing the growth. Now imagine the microRNAs are going to regulate the gene expression. Suppose a microRNA, its target is an oncogene. What will happen to cell proliferation? Can anyone tell me? Huh? No, the target is oncogene, and the function of the microRNA is to suppress that gene.
Proliferation will stop.
So it will be useful for cancer treatment, correct? So if the targets are oncogenes, then such a microRNA will be a tumor suppressor microRNA, correct? Because its microRNA, what is its job? It will shut down the gene expression. If that gene that is shut down is an oncogene, then it is useful for the treatment of cancer, correct? You understand this way, in a simple manner, because it's a public lecture, so I don't want to go into technical details, but for an understanding. Imagine the reverse. I told you one microRNA can have several target genes. If the microRNA's target is a tumor suppressor gene, it will actually help in the development of cancer. It will induce cancer, right? So such a microRNA is called oncogenic microRNA, right? So you call that as oncomis. There are two types in cancer biology. In other fields, there are so many things, but cancer biology, there are certain, to make the long story short, the microRNAs can do both ways. They can induce cancer, they can suppress cancer. It all depends on what their target is. So they can behave as oncogenes and tumor suppressor genes by themselves. So those microRNAs which behave like oncogenes, they are called oncomirs. Those which can behave like a tumor suppressor, they are known as anti-oncomirs or tumor suppressor microRNAs. Just like what you will think. Even some people want to call with fancy names, apatomirs, metastatomirs, and so on. So people have all these kind of names. Anyway, just understand this. So cancer biologists became very interested in knowing about microRNAs because of this reason. because you will be able to regulate the expression of oncogenes and tumor suppressor genes, and this will immediately give a novel treatment strategy. You may ask, sir, why to worry about? You go to a cancer doctor, he will treat either with radiation or with surgery or chemotherapy and all that. But all of you know that one of the major difficulties in cancer therapy is that side effects. Because all these medicines that are given, they will also affect the normal cells. That is why these poor cancer patients sometimes die of medication itself, because that suppresses their immune system, and they die of what are known as opportunistic infections. You and I may not die with a common cold virus, but they may die because they are already treated with so many drugs. I'm just telling, so they are there. The point I am trying to say is their immune system is suppressed. And all of them have side effects. All these three conventional treatments, surgery, chemotherapy, and radiation, all of them have side effects. So there is a tremendous need for identifying you are desperate to cure cancer. So you want to see something new. It is always, the scapegoat or guinea pigs in clinical trials are those who have seen all the medicines and now they are ready to take anything you give, if it can happen. So you need some novel treatment ultimately. So that is why people are looking into. Another interesting thing is, you can ask, sir, this was discovered in 1993 and now these people got Nobel Prize. Is there any medicine out of microRNA? Unfortunately, no. There was only one that went into clinical trials and all that. MIR34 it is called. It's a tumor suppressor microRNA. Somehow some complications are there. They are not able to succeed. But microRNAs, the question asked is whether microRNAs, fundamental work so many people are doing, but have they reached the clinic? The answer is yes or no. Because yes, in the case of certain other disorders, for example, in hepatitis, there is what is called MIR-VIR-SEN something that has reached the clinic almost. Whereas in cancer, it has not reached the clinic. So it takes so many years to, perhaps my gut feeling is these people, in spite of the fact in 2006 itself, the RNA interference people were given Nobel Prize. These people are also given a Nobel Prize for a similar kind of discovery, only because this will encourage others, companies and other people to do more work and find a cure for cancer and similar diseases using these microRNAs. So that is what it is, not still in the clinic. So that is one point I wanted you to understand. So there are only two types of therapies with microRNAs. It's very simple. If the target is oncogene, then this is going to become a tumor suppressor. The problem is, if the another interesting thing is, these microRNAs are also overexpressed in cancer. Mostly such microRNAs are oncomers. So if you take a patient and look out their microRNA profiles, if you see overexpression compared to normal tissue, then such microRNAs are generally called oncomers. So how will you treat such patients? You will have to suppress that microRNA, correct? Because this microRNA, if it is overexpressed, it is actually inducing cancer. I just told you there are two types of microRNAs. Some of them will induce cancer, some of them will suppress cancer. If the patient has more such microRNAs which will induce cancer, you want to inhibit that. That is called microRNA inhibition therapy. The other type, the tumor suppressor microRNAs, they are usually, compared to normal cells, their level of expression, I'm talking about microRNA expression, is very low in cancer patients. So in such cases, you want to introduce that microRNA so that you will be able to cure microRNAs. So that is known as microRNA replacement therapy. So if you are talking about cancer therapy with microRNAs, there are only two types. One is called microRNA inhibition therapy. Another one is called microRNA replacement therapy, depending on what type of microRNA it is. Because it is a double-edged sword. It can induce cancer, it can also cure cancer. So this is why we as cancer biologists are interested in this one. You remember the microRNAs are having their own genes. So whatever problems the genes have, what problems they have? They have a mutation, deletion, insertion, all kind of problems. So those problems can affect the microRNA expression also. That is what is shown in this. And another thing is, the genes can be regulated by epigenetics. I don't know whether you know about epigenetics. Epigenetics means apart from genetics, it will not affect the gene sequence, but it will affect the expression. For example, in the DNA promoter, if there is a methyl group, if there is a methylation, that can, in a particular place in the promoter, that can affect the expression. If that expression of that gene happens to be a microRNA producing gene, then the microRNA expression will come down. So this is the epigenetic regulation. MicroRNA expression, just like any other genes, microRNAs can also be ultimately controlled by epigenetics. So epigenetic alteration. So the genes will have these defects. They will have deletion, mutation, or frame shift mutation, inversion, so many other types of abnormalities. All these things will affect microRNAs also. Yeah, next. This can happen at any level. A microRNA can be produced. The mechanism of microRNA perturbation in cancer what is the title of that particular slide. So it can happen at any stage. Next one. Yes. This is another interesting thing. There is an inverse relationship between microRNA and its target. This is a very interesting thing. If a particular microRNA is highly expressed, what will happen to its target? Target will be low. So you can see in this slide, the microRNA expression in a particular is called temporal switch. If the microRNA level goes up, the target level will come down. There is an inverse relationship between them because it is suppressing the target. It's very simple. Now, this will explain some tissue identity also. Remember, in the beginning of the lecture itself, I told the tissue identity is based on gene expression. So if a microRNA is on, then depending on the tissue and the genes that are present, it may promote cancer or it may suppress cancer. So tissue identity as well as the tissue condition, all these things, modulation of many unrelated targets also is possible. So you can see here, microRNA off means target will be on. If the microRNA is absent, the target expression will go up. If the microRNA expression is high, the target expression will go down. So that is an important thing. I will tell you, there was when we were doing these experiments, next, there was a paper in Nature, this one. So these theories were coming up. So if you have a microRNA that targets, in this case, I think they have a microRNA that will be targeting P10. P10 is a tumor suppressor gene. So if this microRNA, whatever the microRNA is, these people were doing some experiments, they were transfecting that microRNA into cells. The target is P10. P10 is a tumor suppressor. So what will happen to the cells? The target is a tumor suppressor. So if you suppress a suppressor, you will activate. If you inhibit an inhibitor, you will activate. Right? That is the strategy. So here it is a tumor suppressor. So once you put this microRNA, you will expect the cells to proliferate. Surprisingly, the cells started dying. So they didn't understand what was the problem. Then they found out that in this P10, in this particular cell line they were working, It may be different. See, there is another problem. One microRNA may work as an oncogene in, let us say, in breast cancer. In another cancer, it may work as a opposite. In one tissue type, it can work as an oncogene. In another tissue type, it may work as a tumor suppressor gene. So these kind of variations are there. That is why in the clinic, if you are going to use them, you have to use them very wisely. Unless you know the basics, you will not be able to take it to the clinic. Unless you know everything about that. If your foundation is not strong, definitely you are going to face with this. Happened. Many medicines, not in this field, but in many other fields, many drugs have been marketed by big companies and only to be withdrawn because of the side effects afterwards. If you don't do basic science properly, if preclinical trials, preclinical experiments have not been carried out properly, then you will face all these problems, litigations and withdrawal of the medicine and so on. That target. So here, I'm sorry, I'm dragging too much. No, go back to that. Now, they were surprised why this P10 is expressed and why the cells are dying. P10 is not suppressed. Then they found that in this particular cell line, that P10 mRNA UTR was something like a pseudogene. You heard of pseudogene? It was not doing, that copy number of that pseudogene was excess than the normal wild type gene. So it was able to bind with the microRNA and nullify its effect. So the microRNA was actually ineffective. Since the cell line expressed P10, the cells were dying. So this was that they call as decoy for microRNAs. They said that these sequences which are ineffective, the 3' UTR, they called them as decoy for microRNA. This has got a clinical implication because if you have those sequences, you can use that as attacking for microRNAs. Suppose you want to take out microRNAs from the cell, you want to have these sequences, put them onto cells, they will deplete these microRNAs because you don't want this microRNA because it is inducing cancer, for example. So that was one of the strategies. Next. So as I told you, I'm coming back to my original thing. In colorectal cancer, people have found several sequence of events, how this cancer develops, thanks to Bert Vogelstein. His model of colorectal pathogenesis is well known. So microRNAs were found to be involved in almost every stage of that. There are several stages. I don't want to explain that in detail. So next. Now I will come back to, this is what the strategies are: microRNA inhibition and microRNA replacement. By using these strategies, people have found that these microRNAs can be used for treating people with resistance, chemoresistance. You can make them sensitive. More than that, you can also detect certain cancer subtypes by looking at the microRNA profiles of people. Remember, genetic profile people are doing. Similarly, if you do microRNA profile in the blood and other tissues, you may be able to classify the subtypes of cancer. You can identify. So that is known as signature. In these areas, diagnostic purposes and prognostic purposes, some improvement has happened. People have, some progress has been made in the on the research side. I told you there was a failure in ultimately taking it to the clinic. There were issues. There are issues still continuing. But on the other side, for diagnostic purposes and for prognostic purposes, there is a lot of scope on this. Next. Yeah, these are some of the strategies people use. I told you, if you can make some sequences which can take away the microRNA from the cell, they are known as sponges. Because in the sponge, what we do? A sponge, women must be knowing, like in the kitchen, our mothers, sisters, they do it. They put the sponge, whenever there is a spill of water, the sponge absorbs water. Similarly, if you think this microRNA is a spillage, then put that sequence that will bind that in excess, so that the microRNA can be depleted from the cell. Such a technology is known as sponge technology. They do it. Similarly, if you want to introduce the see, I told you there are only two things. Either you inhibit microRNA or you introduce microRNA for treatment of cancer. If you want to introduce, you introduce similar sequences of microRNA and introduce them into the cell. That's known as microRNA mimics. So either you introduce mimics or you create sponges so that you can deplete. There are many, many other methods. You can introduce anti-mirs. You can also introduce siRNAs. Many things can be done. Viral expression vectors, small molecules, that is also coming up. Certain chemicals which will inhibit the production of these microRNAs, because after all, they are also produced from their own genes. If that original gene can be inhibited, the microRNA synthesis can be inhibited. So those kind of things are being tried. Next. Yeah, this is also like several strategies. Next. There are many companies which are, go to next because it's the same thing. This also kind of I told, so go to, yeah. There are many companies in the world who are interested. Now with this Nobel Prize, more and more companies will come. Prominent among them are miRNA Therapeutics, Miragene, Anilam. So many companies in the US are very interested in microRNA. See this, they have to invest millions of dollars. Ultimately, for the past 10-15 years, it has not gone to the clinic. So there is a risk involved. But then it is still an attractive method because it can attack so many genes. For example, one way to look at it is cancer is a kind of disease in which so many genes are affected. So you cannot cure it by affecting only one gene. Of course, you can in certain cases, exception, what we call oncogenic addiction. If you are addicted to one gene, then if you attack that gene, you can cure. That is an exception. But generally speaking, multi-genes are involved. So if you can affect with something that will affect many genes at the same time, perhaps that here it will do, because one microRNA has 500 genes as its target. So in this way, people are trying. Let us see. Next. These are the implications I already told you. That is, preclinical trial research is to fully clarify these aspects before it can be taken to clinical applications. Next. Okay. You may ask, sir, what have you done in your lab? We have done many microRNAs. We have found many microRNAs which can, for example, in colorectal cancer, somebody said whether I have done with breast cancer. Actually, this work only I have done with breast cancer. In my postdoc, I have done something with breast cancer. Other than that, I am not really an expert. My colleagues are there who are working on breast cancer. Most of my work was on either cervical cancer or on colon cancer and so on. It was not really cancer specific. It was more like signaling specific. So that is what I did a little bit. In this particular work, it is on breast cancer. I will tell you why we took up this. You remember I told you that you can go, if you know some gene, you can go back and find out the microRNA. That is what we did in this one. Yeah, go to the next one. I think you may be knowing what is called apoptosis, cell death. See, what happens is the cells, if you, for example, the famous example we give is that if you attack the cells with radiation, there will be DNA damage. And if the DNA damage is small, the threshold is small, then the cells will try to repair, which we call as DNA repair mechanisms. So the DNA, what is DNA repair mechanism? DNA structure is altered. You put back that to original structure. Whatever does that is a DNA repair mechanism. That is as simple as that. Repairing is what? Restoring the original structure. There is some damage and it is restored to the original structure. Now, if the damage is small, they will repair. But if it crosses a certain threshold, they will die. There are different forms of cell death. One form of cell death is called necrosis, where it will completely die, and there will be there are certain differences. But in many cases, almost in all mammalian cells have this capacity to die themselves. If the signal is such that they cannot tolerate that anymore, they will kind of commit suicide. It is an interesting thing. Necrosis is akin to murder. In murder, it is somebody else decides that you have to die. But in suicide, you are deciding that you have to die. Here, that is why it is akin to apoptosis, but don't stretch on that. These examples are given just to make you understand. So it is like suicide. Cellular suicide is what is called apoptosis. When the signals go beyond a certain threshold, the normal cells, they will commit suicide. That is called apoptosis. Now, why I am saying this is because the cancer cells, they don't obey this rule. Normal cells, they will die of apoptosis. Cancer cells, they will not die. They want to survive. So they will not die under these conditions in which normal cells would commit apoptosis. So in other words, cancer cells will evade apoptosis. Cancer cells are resistant to apoptotic mechanisms, usually. Like I told you, there are factors which will induce apoptosis, which we call as pro-apoptotic factors. There are factors which will oppose apoptosis, which are known as anti-apoptotic factors. In the cancer cells, there will be anti-apoptotic factors. Whereas if you introduce pro-apoptotic factors, you may be able to kill them. There is a balance between the two, and that is also one of the mechanisms. It's not the only one mechanism. So microRNAs also were shown to induce apoptosis. So we were interested in that actually. So we tried to look at some microRNAs. And in this case, we looked at what is called SLC7A11. For simplicity, you can take it as it's a survival protein. That is, it will offer, it is anti-apoptotic. It will resist apoptosis. It will give survival advantage to the cancer cells, for example. So it will not allow the cancer cells to die. So what you have to do if you want to treat cancer, there are many cancers in which this protein is supposed to be in high concentration, which means if you put it down, if you suppress its expression, then cancer cells will die. Question is, can you do it with some microRNA? Because microRNA will suppress the expression. In this case, SLC7A11. Next. That's what we tried. Interestingly, let us not go into the details. Yeah, go to the next. SLC7A11 is supposed to be overexpressed in many cancers, upregulated, as the slide shows, in lung cancer, breast cancer, glioblastoma, ovarian cancer, and all that. Next. At this time, when I was looking at this apoptosis, a student from my lab came and told me, sir, there's a recent paper on ferroptosis. This is a new form of cell death. Whoa. By the way, my own teaching philosophy is that teacher is somewhat a wrong word, because teacher is basically a learner. He learns all the time. So I learned from the students only. The student came and told ferroptosis. That's the first time I heard about ferroptosis. I didn't know. Then he said, that's a new form of cell death. Why don't we work on that? And I said, yeah, it looks very interesting. Why don't we look at it? That is how we started this work. Unfortunately or fortunately, I don't know who is unfortunate. The student left the lab. I told him, if you show a microRNA inducing ferroptosis, that's a new form of cell death, we can publish it in Nature. With that enthusiasm, I started motivating that boy. And I also got very excited. Unfortunate things didn't work that way. He left the lab. The funds were not coming. I had a few crores of funds, but in the third year, the funding didn't come. So I couldn't complete that project. We did that and we published. But unless you are very quick, there is a cutthroat competition if you want to be at an international standard. So we did it finally, but then by that time, many people also published. We couldn't do it quickly. Only if I had money and other resources. Even sitting in IIT, I'm telling this. Yes. So, so it is very difficult to be internationally competitive. Although we have the technology, we have the knowledge, everything, this is one of the problems. Interestingly, today morning I read same things written by these Nobel laureates. That is what Victor Ambrose himself says. When he discovered these things, there are 3-4 labs which were doing the same thing. And he and his wife were stunned by that because they thought nobody would discover microRNAs in nematodes. They were the first ones, but that was not the case. There were many people. So they were forced to submit the paper to Nature or Cell in a hurry without completing. This is the situation. When it is very competitive, it is very, if you are in a competitive area, this is one of the problems. Finally, anyway, I keep on talking. Yeah, next. So ferroptosis is a new form of cell death where it involves, it is similar to apoptosis, but only different, one small difference. There are many differences, but main difference, as the name implies, ferro means iron. So it requires iron for the death, right? So iron dependent, I don't know whether we can say apoptosis, we can say cell death, iron dependent cell death, we can say. So it requires glutamine being lysed, so it is glutaminolysis and so on. Lipid ROS, the major killer in this, what is the killer about with this ferroptosis? The killer is reactive oxygen species, especially lipid ROS, as we call. Lipid reactive oxygen species is the main killer through ferroptosis. So iron is required and the killing mechanism is through lipid ROS. So this is what he told. There are many forms of cell death: apoptosis, paroptosis, necrosis, necroptosis, all kind of names are coined. There may be some small differences. Yes, I'm going to stop here. This is what we did. We finally found that by inhibiting this SLC7A11, we were able to kill the cancer cells, breast cancer cells. We were also able to show that it doesn't metastasize. So there are so many figures. You can read that. It came in the cancer letters. I will give you the reference. Can you go quickly to everything? Because there is no time. I already exceeded the time. So, yeah, go just to keep moving. I told about the hypothesis. The results, of course, I cannot describe everything. Yeah. Maybe we will go to the conclusion and just say what it is. Can you go to the last slide? The problem is it's a public lecture, so I cannot go into all the technical details. Otherwise, of course, yeah, just before that. Yeah. No, before that. One slide before this. Yes, yes. Here, if you touch, it will come one by one. It is not coming in the conclusions. Yes, if you touch, yeah, just, yeah, stop. Yeah. Now, we showed that MIR5096 regulates the SLC7A11, but like I said, 3' UTR, We have shown that. And we also showed that it induces ferrotosis by increasing the ion accumulation, hydroxyl radicals, reactive oxygen species, lipid ROS, and mitochondrial membrane potential. And we express this microRNA, and we showed tumor suppressive properties for this microRNA. The way we found was SLC7A11 was taken, this 3' UTR was examined, and we found some microRNAs. We picked up this microRNA 5096 because it had a very good sequence complementarity, and it worked. Remember, in the beginning when I started in 2006, such a methodology didn't work, but here it worked. So it mediated, and that is what we have shown, that it is a tumor suppressor microRNA. For those of you who want to read, you can look at it. It came in some. Can you go to the last one? Go, not next. Yes, this is what we published in Cancer Letters. It's a very good journal, and somebody highlighted it in some newspaper also here in Chennai. So I think I will stop here. Yeah.
Thank you very much, Professor. It was a wonderful lecture, and many of those who don't have biology background has learned a lot from this lecture. It's very, very simple, and we enjoy the lecture. So now it is a question answer session. Those who have any question, please raise your hand. We'll come to your place and give you the mic. Before that, there's going to be a small memento given by the Anna Centenary Library.
இருந்தாலும் கொஞ்சம் சில டைம்ல 40 வருஷம் இங்கிலீஷ்ல
Sir, you need to answer.
Yeah, we can do it. Yeah, if you have any. Yeah, this
Sir,
one.
why cancer kills the people?
Basically, I'm not a doctor, but why it kills the people, you are saying?
Yes, sir.
Because it affects many genes, 300 genes and so on. So you need these genes to be active. You want to be active. So ultimately, it interferes with your metabolism. So you die. Yeah, all the systems are crippled means what will you do? So that is a very general, very vague answer, but it depends on, and there are 200 types of cancers. So you will have to look at why it is. Sometimes it is due to some oncogenes. Overactivity. The cells proliferate and you get tumors and they block. So it depends on the place. For example, even if something is not a tumor, depends on the place. In the brain, which may be what are known as benign tumors. They are usually benign tumors are harmless. Depends on where you get it. In the brain, if you get them, the very fact that they bulge and then press the brain tissue, brain is such an important thing. If you press some part of the brain, you may lose, maybe you will become paralyzed. Depends on where you touch. And if you put the pressure, mechanical pressure, there is no real disorder there. Just only you are pressing. You may have all kind of effects. Depends. The biology is like this Jekyll and Hyde. The same person is different in the night and in the daytime. He is a good person, right? Same thing, something you call it as an oncogene, for instance. You always think, because mechanistically we are thinking like that. Oncogene means it should be growth promoting. It's not true. If you give that oncogene is overexpressed in some cell, instead of growing, they will kill the cells. Experimentally also we have seen. If you introduce that oncogene in excess, அளவுக்கு மிஞ்சினா அமுதம் விஷம் சொல்றோம் இல்லையா அந்த மாதிரி sometimes it can do the opposite also. Anyway, let me not keep talking. Yeah.
So I have two questions.
Yes.
The first is related to your lecture.
Yes.
You said that the RISC binds to the 3' UTR region.
What is that?
The 3' UTR region.
Which one binds?
RISC.
What is RAC?
RNA-induced silencing complex.
No, it is a complex. It doesn't, it has proteins where the RNA can go and bind. That is what I said.
Yes, sir, but the UTR region is untranslated. Like, how does it affect?
What?
The UTR region, it's untranslated. So how does it affect?
You should know what is UTR. UTR is a part of your messenger RNA, correct?
Yes.
In the front, you know the numbering. It starts from 5' to 3'.
Yeah.
So it will be in the 3' in this
Yes,
case,
sir.
3' untranslated region. So there it goes and binds. That is what I said. What is your question?
UTR stands for untranslated region, right, sir? So we
Yeah,
can't
that
translate.
UTR means you are referring to RNA, not protein. Yeah, now you.
So it's
Risk is different. Don't confuse with the risk. Risk is a general name given to many components in the cell which are associated with this RNA and other things. You understand? Organoid proteins. It's a complex thing. Cell, we are, for our understanding, we are calling something as carbohydrate, protein, nucleic acid, lipid, and so on. Okay. But in the cell, if you take, for example, it is very difficult to isolate a protein from the cell in a pure form because it will not be a pure protein. It will be a glycoprotein, lipoprotein, nucleoprotein. That means they always live together. They don't want to get isolated. They are always together, all these molecules, to separate, even covalently bound. So it is very difficult to separate them. Similarly, this risk is also a complex. It has many other things. You are isolating yourself into 3' UTR.
oh, okay.
That is the problem. It is a complex system and it contains many other partners, not only 3' UTR.
Sir, the second question.
Yeah, sure.
What is so special about microRNA which sets it apart from siRNA? What's the difference in how they work?
Yeah. siRNA, you have a complete sequence complementarity, right? So it has to be, and it will, there will be a complementarity. But here, partial complementarity and so on. There are many other small differences, but it is sufficient to understand that way.
Thank you, sir.
Also, in the gene coding regions, you are usually targeting.
Yes,
In the
sir.
case of siRNA. Here it can come from introns also, if you know the difference between introns and exons. So that is also one difference. There are many other differences.
One question came from the YouTube.
He's asking. Yeah. Ask.
So, can we dock anti-cancer protein plus miRNA in vesicles as potential drug?
Yeah, that is coming up. I mentioned that actually in the exosomes and all that. They are also vesicles.
Hello, sir. Good evening, sir. So thank you for the nice presentation. So my question is, you said two types of micro mRNA. So the one is onco micro mRNA which incubate the oncogene.
Don't just say microRNA.
Yeah, sir.
Don't say
MicroRNA.
micro mRNA. You are
Okay.
mixing up the target and it doesn't matter. I'm not
Okay, sir.
criticizing. I'm just saying.
Yes, sir. So the miRNA incubate the oncogene which will not promote the cancer. So the first type, and the second type, it will bind with tumor suppressor gene. So it will induce the tumor suppressor gene and it will kill the cancer. So what are the structure difference between two type of miRNAs?
There is no difference in the structure of the miRNAs. It's not that.
Like, how should we identify that?
Just wait. It is not about the microRNA. It depends on what it silences.
Okay, sir.
That's all. If you are silencing an oncogene, you are a tumor suppressor. If you are silencing a tumor suppressor, if you suppress a suppressor, you become an activator. If you are suppressing a tumor suppressor, you are activating the tumor. Understand like that. It's easy.
Okay. Thank you, sir.
So, same person can do both. That is why I said it doesn't depend on the microRNA. Let us imagine I am the microRNA, right? I can sometimes act as an oncogene. Sometimes I can act as a tumor. That's why I said Jekyll and Hyde. Same person, I may be good, I may be bad. Night I am bad, daytime I am good. So same way.
So are there any anti-cancer drugs which regulate the mRNA?
But what?
Any anti-cancer drugs?
That's what I just told you. It has not seen the clinic yet. There is one which is called a microRNA 34. It is being tried, but people are working. Still, there is no actual treatment strategies are there, but successful strategies are not there for cancer, for treating cancer. Still, it has not reached the clinic.
Okay.
But there are many people, many ways they are trying. Many methodologies I have shown.
Yeah.
Sponge, mimics, antimers, LNAs, so many methodologies, liposomes, so many methodologies are the nanotechnology, so many things are being tried. But nothing has come to the clinic so far in the treatment of cancer. But in other diseases, they are finding
So
some
how about
success.
signaling?
Huh?
Supposing some anti-cancer drugs acting on the signaling pathway. Are there any
Sir, the
drugs?
problem is, it is not one thing that is happening. Suppose it's only one signaling and if you shut down, you can cure cancer means it is okay. But 300 means what will you do? So that is a problem.
Yes, sir. Thank you, sir.
Sir, what is the role of miRNA in stem cell differentiate into many cells? What is the role of microRNA in stem cells differentiating into many cells? You are saying
Stem cells differentiating into what?
அதாவது செல் வந்து பல செல்லா மாறுது அதுல வந்து மைக்ரோ ஆர்என்ஏ முக்கிய பங்காற்றுதுன்னு சொன்னீங்கல்ல சார் அது கேட்கிறேன் அது வாட் இஸ் த ரோல் ஆஃப் மைக்ரோ ஆர்என்ஏ
மைக்ரோ ஆர்என்ஏவோட ரோல் வந்து ஜீனோட எக்ஸ்பிரஷனை சப்ரஸ் பண்றது. நீங்க சொல்ற ப்ராசஸ்ல ஜீன் எக்ஸ்பிரஷன் சேஞ்ச் ஆச்சுன்னா மைக்ரோ ஆர்என்ஏ ஒரு ரோல் இருக்கும் அதுல. அது எப்படி இருக்கும்னு இப்ப நம்ம சொல்ல முடியாது. அதுல வந்து அந்த ஜீன்ஸ் என்ன பண்ணுது அப்படின்றத பார்க்கணும். சப்போஸ் ஒரு பர்டிகுலர் டைப் ஆஃப் ஜீன்ஸ் வந்து நீங்க சொல்ற மாதிரி ஒரு செல்ல இன்னொரு செல்ல மாத்துதுன்னு வச்சுக்கோங்க, ரைட்டா?
நீங்க ஸ்டார்டிங்ல சொன்னீங்க.
ஆமா ஆமா ஆமா, அதெல்லாம் ஒரே ஒரு ஜீன் பண்ற வேலை இல்லை. அங்க ஆயிரக்கணக்கான ஜீன் இருக்கும். நீங்க எந்த மைக்ரோ ஆர்என்ஏ பத்தி பேசுறீங்க, எந்த ஜீனை பத்தி பேசுறீங்கன்றத பொறுத்து தான் ஆன்சர் சொல்ல முடியும். அதனால நீங்க எப்படி கேக்குறீங்களோ அத மாதிரி நான் பதில் சொல்றேன்.
ஓகே, மைக்ரோ ஆர்என்ஏ வந்து சப்ரஸர் ஜீன் சொன்னீங்க, சப்ரஸ் பண்ணுது. தட் இஸ் அது வந்து ஏதோ ஒன்னு அழிக்கிறதுன்னு சொல்றீங்கல்ல சார், அந்த அழிக்கிறது எப்படி டிஃபரன்சியேஷன்ல யூஸ் ஆகுதுன்னு கேட்க. அந்த செல் வந்து ஸ்டெம் செல் வந்து பல செல்லா மாறுதுன்னு சொன்னீங்கல்ல, ஃபைப்ரோபிளாஸ்டா மாறுது, எபிதெலியல் செல்லா மாறுது, அந்த மாதிரி சொல்றீங்கல்ல.
நான் உங்களுக்கு என்ன பதில் சொல்றேன்னா, அந்த டிஃபரன்சியேஷன் நடக்குது இல்லையா, அதை ப்ரமோட் பண்ணாலும் பண்ணும், அதை பிளாக் பண்ணாலும் பண்ணும். அதுதான் சொல்றேன். ரெண்டு வழியும் செய்யும். டபுள் எட்ஜ்டு நைஃப், நல்லதும் செய்யும் கெட்டதும் செய்யும், கடவுள் மாதிரி. ஆனா நீங்க இப்ப அதுக்கு பதில் சொல்ல முடியுமா? நமக்கு ஏன் நல்லது நடக்குது, நமக்கு ஏன் கெட்டது நடக்குதுன்னு சொல்ல முடியுமா? சொல்ல முடியாது. சூழ்நிலையை பொறுத்து இருக்கு. அது மாதிரி தான்.
அது அந்த இடத்துல எப்படி டிஃபரன்சியேஷனுக்கு அது எப்படி யூஸ் ஆகுதுன்ற மாதிரி
டெக்னிக்கலா போனா தான்
ஓகே ஓகே, நான் சொல்றேன் உங்களுக்கு, அதுக்கு நல்ல பதிலே சொல்றேன் உங்களுக்கு. அது எப்படின்னா, கேன்சர் செல்ஸ் ஆர் புவர்லி டிஃபரன்ஷியேட்டட். புரிஞ்சுங்களா? கேன்சர் செல்ஸ்ங்கிறது வந்து டிஃபரன்ஷியேஷன் கம்மியா இருக்கும், அப்படி வச்சுக்கணும். அப்போ நீங்க வந்து அந்த டிஃபரன்ஷியேஷனை இன்க்ரீஸ் பண்ணீங்கன்னா அது ஒரு ஸ்ட்ராட்டஜி டு கியூர் கேன்சர். ஒரு பர்டிகுலர் இது கூட ஆட்ரான்னு சொல்லுவாங்க, என்ன சொல்றது டக்குன்னு பேர், அந்த ரெட்டினாயிக் ஆசிட் தெரப்பி இருக்கு. அந்த தெரப்பியில வந்து நீங்க இன்டியூஸ் பண்ணீங்கன்னா, டிஃபரன்ஷியேஷன் இன்டியூஸ் பண்ணீங்கன்னா அந்த பர்டிகுலர் டைப் ஆஃப் கேன்சர் கியூர் பண்ணும். ஸோ அந்த டிஃபரன்ஷியேஷனை ஒரு மைக்ரோ ஆர்என்ஏ இன்டியூஸ் பண்ணிச்சுன்னா இட் வில் ஹெல்ப் ட்ரீட்டிங் கேன்சர். அது இருக்கு, தெரப்பியே இருக்கு. Yeah.
What you are saying is a cell that is a totipotent, it can go into a brain cell or your bone marrow or your tissue or muscle cell like that. Maybe that microRNA may inhibit one thing. So you can, if you want to put it as if you develop into a brain cell, it may develop into a brain cell. And if it inhibit one thing, and it may become a muscle cell. Maybe that may be the answer. Okay, understood?
See, the problem with that kind of thing is there may be many microRNAs, many genes are involved. You cannot just answer with something like that. No, that's what. Yeah. But he was specifically telling about differentiation. There is a therapy with ATRA. That's what I told you, retinoic acid. So I'm not getting the full form of it. Anybody can tell? I forgot. Okay. Yeah. Yeah.
So I'm not from biology background. So
Doesn't matter.
I'm working as an ML engineer. So if I can contribute, what should be the problem statements I should look into in order to suppress the engineers? I mean, scientists are trying to fix some problems here, right? So what should be the ideal problem statement that ML engineers should look on to exactly? I'm not sure.
What you are asking is, what is the problem? Why it is not seeing the clinic? That's what you are
I'm
asking.
not sure. Actually, not that. You're saying there are too many kinds of patents right here.
Too many types of
each
what?
patents for each
What?
representations of this.
Representation of what?
What? So this what's the direction?
miRNA.
mRNA. So
Not mRNA, miRNA. MicroRNA. Say like,
So
yeah.
we are trying to find out microRNA for each type of representations on each cell site. So is it feasible? Is it easy to find that one for every kind of cells in the human body, or can ML contribute in it somehow? That's what I'm trying to ask.
See, all the tissues may have microRNAs. We don't know what is their role. If you remember, in one tissue it may act as an oncogene. In another tissue it may act as a tumor suppressor gene. So it is complicated. Unless you study that in detail, you will not be able to say that. Same thing. This is not only about microRNAs. Many biological molecules are like this. It's not about microRNAs. That's why biology is complicated. Yeah. Yeah. Kalan.
சார் இப்போ கேன்சர் ட்ரீட்மெண்ட்ல ஜீன் தெரப்பின்ட்டு நிறைய நடக்குது இல்லை சார்? இப்போ நான் எக்ஸாம்பிள் ஒரு லங் கேன்சர் எடுத்துப்போம் சார். இப்போ லங் கேன்சருக்கு நான் வந்து ஒரு ஜீன் தெரப்பி கொடுக்குறேன் சார். மேபி இது வந்து என்னன்னா இன்னொரு டைப் ஆஃப் செல்லையும் இன்டியூஸ் பண்ண முடியுமா சார்?
இதுல என்னன்னா, இந்த ஜீன் தெரப்பியில ஒரு ஃபேமஸ் கோட்டிங் இருக்கு. நம்ம இந்தியன் தான் அவரை, இந்தர் வர்மான்ட்டு. இஸ் அ பிக் எக்ஸ்பர்ட் ஆன் ஜீன் தெரப்பி. நல்லா லென்டிவைரல் தெரப்பிலாம் அவர் தான் நிறைய கண்டுபிடிச்சிருக்காரு, எல்லாம் பண்ணியிருக்காரு அவரை. அவர் வந்து ஒரு 20 வருஷத்துக்கு முன்னாடியே வந்து ஒரு கோட் சொன்னார். எப்படி சொன்னாருன்னா, நமக்கு இப்போ மாலிகுலர் பயாலஜி இவ்வளோ டெவலப் ஆயிடுச்சு, அவன் குளோனே பண்ணுறான், மனுஷனே உற்பத்தி பண்ணுறான், அது இதுன்னு சொல்கிறாங்க, எல்லாம் சொல்கிறாங்க. டெக்னாலஜி இஸ் அட்வான்ஸ், நோ டவுட். பட் இந்த ஒரு ஜீனை வந்து ஒரு பர்டிகுலர் பிளேஸில் இந்த மாதிரி ஒரு டிஷ்யூவில் கொண்டு போய் சேர்க்கறதுக்கு கரெக்டான டெக்னாலஜிஸ் இல்லை. புரியுதுங்களா? அதனால தான் அவர் சொன்னாரு, there are 3 important problems in gene therapy. என்ன 3னா டெலிவரி அப்படின்னு சொன்னார் ஒரு டாக். அதுதான் டெலிவரி இஸ் அ ப்ராப்ளம். டெலிவரி அட் அ ஸ்பெசிஃபிக் சைட் இஸ் நாட் தட் ஈஸி as we think.
சார் இப்போ லைக் ரேடியேஷன் தெரப்பி சொன்னீங்களா சார்?
Yes.
இப்போ வந்து என்னன்னா அது வந்து பார்ஷியலி இப்போ ஒரு டிஎன்ஏ வந்து டேமேஜ் பண்ணுதுன்னா மேபி அதுவே வந்து கேன்சரை இன்டியூஸ் பண்ணுவோம்.
Yes, it's a double-edged sword. Radiation can be used to treat cancer, can induce cancer also. அதான் சொன்னேன், அமிர்தமும் விஷமும் சொன்னேன். அமிர்தம் வந்து நல்லது தான். ஏன்னா எக்ஸஸாக சாப்பிட்டீங்கன்னா ஃபுட்டே வந்து பாய்சன் தான், நிறைய சாப்பிட்டீங்கன்னா. அந்த மாதிரி தான் எல்லாத்துக்கும் ஒரு அளவு இருக்கு, டைம், டோஸ் எல்லாம் இருக்கு. அதே மாதிரி தான் இங்கேயும்.
இப்போ ஜெனட்டிக் இப்போ ஒரு பேபி வந்து ஜெனட்டிக் அப்நார்மலிட்டியா பிறக்கிறாங்கன்னா அதுக்குமே இப்போ நிறைய எம்ஆர்என்ஏ வந்து இன்னும் கொஞ்சம் டெவலப் பண்ணா அதையும் நம்ம கியூர் பண்ணலாம் சார்.
ஜீன்ஸ் இன்வால்வ் ஆகிற எந்த ஒரு விஷயத்துக்கும் எம்ஐஆர்என்ஏஸும் ஹெல்ப்ஃபுல்லாக இருக்கும். ஆனால் வந்து இப்போ இதெல்லாம் வந்துருச்சான்னு கேட்டால் வரலன்னு சொன்னேன். நான் தான் கிளினிக்கில் வரல, ஒன்லி ஹெப்படைட்டிஸ் சி மட்டும்தான் வந்திருக்கு, கிளினிக்கில் வந்திருக்கு. கிளினிக்கில் வர்றதுங்கிறது வந்து நம்ம சொல்லலாம் ஈஸியாக, பட் அது வந்து டைம் ஆகும். சைடு எஃபெக்ட்ஸ்லேருந்து நிறைய விஷயங்கள் இருக்குது. அதை பற்றி ஃபஸ்ட்டு நமக்கு நல்ல நாலேஜ் இருக்கணும். அதெல்லாம் ஸ்பெண்ட் பண்ணால் தான். இப்போ எய்ட்ஸ் வரும்போது அதுக்கு க்யூரே இல்லைன்னு சொல்லிட்டு எல்லாம் பயந்துட்டாங்க. ஆனால் அதுக்கு முன்னாடி வைரஸை பற்றி நிறைய பேர் படிச்சிருந்ததுனால தான் அதுக்கு ஒரு மெடிசன் கண்டுபிடிக்க முடிஞ்சுது ஷார்ட் டைமில். ஸோ அதான் சொல்லுவாங்க, பேசிக் சயின்ஸும் அப்ளைட் சயின்ஸும் எது இம்பார்ட்டன்ட்னு சொன்னால் தேர் மஸ்ட், அப்படிலாம் இல்லை, தேர் மஸ்ட் பி சயின்ஸ் டு அப்ளை அப்படின்னு சொல்லுவாங்க. இவர் சம் ஜெர்மன் மார்க்ஸ் பிளாங்க் சொன்னார் அது மாதிரி. ஸோ எல்லாத்துக்குமே வந்து உங்களுக்கு பேசிஸும் வேணும். ரெண்டுமே இம்பார்ட்டன்ட் தான். தேர் லைக் போத் சைட்ஸ் ஆஃப் த காயின் அப்படின்னு சொல்லலாம். எது இம்பார்ட்டன்ட்னு இல்லை, பட் ரெண்டும் வேணும். ஒன்றும் இல்லைன்னா ஒன்றும் இல்லை. அதுதான் ரைட்.
Thank you. It is the time to wind up. I request Aditya Verma, Joint Secretary of PSL, to deliver the vote of thanks.
Good evening, all. So before going to my duties, I know you will have a lot of questions. So in the feedback form, you can add your question. We will try to compile those questions and send to sir and try to get an answer to that. Since you are all registered, your email ID will be added to a Google group. We will spread the answers through that. So please, if any you have further questions, please raise in the feedback form. There is a specific area is there. So with this, sir, thank you very much for your, thought-provoking lecture. He has gone through the history of, microRNA. And one who is involved in that research, so it is more effective. From his own experience, he talked about the Nobel Prize of this year for physiology and medicine. That was added advantage for us. So thank you for your lecture, and thank you the Anna Centennial Library for arranging this auditorium. Earlier it was planned at 5th floor. It cannot accommodate this much of people. Thank you for all of you to turn out in large numbers. So tomorrow we have a lecture on physics, Nobel Prize for Physics 2024 at 4 o'clock. It will be either at 5th floor here on. Okay. Thank you so much. Good night.